students, welcome to PNYC. So today we are going to study the 8th chapter in your biology text, that is science book, that is cell biology and biotechnology. So to begin with, let us have a brief know-how of what all techniques have we learned so far. So we have initially seen certain chapters, life processes. So all of them, when they are put into certain human use, anthropological use for the betterment of mankind or the animals around. I mean, everything is eccentric to humans. So animal husbandry, so on and so forth. We term it as applications of cell biology and in applications of biotechnology. So this has been discussed in great detail in this chapter. A very interesting and scoring chapter. Most of it is based on examples. You will find even if you have to write short notes or brief answers, the thing is everything remember with an example, as I had told you during the initial phases where we began classes, and then your life is going to be easy peasy. So, do you remember something which pertains to cell biology that we have studied? Recall the earlier chapters. Do you remember what is meant by cell culture, PTC, ATC, right? Plant tissue culture, animal cell culture, animal tissue culture, how do you do? What are the media that we use for cultivating them? What are the methods we utilize for growing and isolating them? So on and so forth. So this is just a build-up of what we have known a little bit, but with certain furbished details. So in this chapter, the 4 major points that we'll be focusing on is your cytology or cell biology, the stem cell biology that is there in great details as an initiatory phase in this chapter. Then we have biotechnology and its applications, and also certain important stages in agriculture development in India as well as worldwide. But obviously, since it's better to know what is there, what you have first. So there are a lot of good examples given about how the Indian government then had revolutionized it with certain revolutions like the Green Revolution, White Revolution, etc. So that in details I will be coming towards the end of this chapter. So to talk about stem cells, a lot of it is being talked about stem cells run at TEDx talks. If you YouTube, you should, if you are aiming to do a career in sciences, listen to certain researchers on TEDx. Okay. So stem cells are what? Stem cells are the cells which are active up till 8 to 16 cell stages in the zygote developmental regimen or the stage. So during embryonic development, when we say that a sperm and egg fuse to form a zygote and embryo ultimately, so up to the stage in which the mass of cells is 8 or 16. Typically, it is when you study developmental biology in details, you will be studying that as well. So you will be studying what is a blastocyst, gastocyst, so on and so forth. So the gastomere stage or the blastomere stage. And then during the blastomere stage, up till that stage, the cells are truly stem cells. They are pluripotent, okay, or they are totipotent, as we call in plant cells. So plant cells are totipotent. Any cell even can differentiate into any kind of cells. Okay. So that is due to their potential of totipotency. So in humans, only at this limited stage, very early in the stage of development, it is called as, or it is capable of being totipotent, not otherwise, or pluripotent. So until the blastocyst stage, that is the 8 or 16 cell stage, it is said to be a stem cell, and the quality is very much advantageous in terms of its pluripotency. So given the right conditions, it could differentiate into any cell. Let it be a brain cell like a neuron, or your interstitial cells, or your intestinal cells, or the cornea of your eye. You give them the stimulus and the environment accordingly, they will develop into those organs or organ tissues. So they have a great and huge potential for development. So what are the sources for these? It is the umbilical cord. Okay, umbilical cord is the cord through which the baby child in the womb is attached to the mother, and that is even present after birth. So that is cut. The umbilical cord is cut. Little bit of ancient knowledge. So usually the umbilical cord was dried and stored. So even up till our generation, if you had grandparents alive or around during the delivery, the umbilical cord was preserved. There are a lot of medicinal applications to it with reference to Ayurveda or certain natural practicing natural medicine practicing regimens. So that was stored. But now due to modern day technology, even the umbilical cord can be preserved And take it from me, students, the cost is like once in a lifetime, I believe, but it is huge. So the only intention over here is to store them in times of need. So when you are say 50 years, 30 years, or any lifetime year, you can just put your finger on its date. If you meet with a mishap, a major accident, which leads to a loss of limb, this and that, so it is believed that due to advent of modern day technology, we will be able to regenerate the organ to a certain extent or a full extent. Right? So that is why these umbilical cords are still preserved. In earlier times, they used to be dried and stored just to for the purpose of certain medicinal use. The cells in it are going to dehydrate and die. But now, due to modern day technology, extreme low temperature storages like liquid nitrogen, right, where the temperatures can be as low as minus 135 to minus 196 degrees Celsius. So these cells can be permanently preserved for an infinite amount of time. So this is an application of biotech that I will be reiterating when we see that heading in your chapter as well. So the third source is your red bone marrow and the adipose connective tissue. Adipose means something pertaining to the fat. So that also is going to contain your stem cells. Apart from that, your bone marrow. So bone marrow is what? The middle hollow cavity of your bone which synthesizes the cells, which synthesizes your RBCs, your white blood cells, right? So that is also going to contain your stem cells. So on certain occasions, surgically, the region from the bone marrow is removed in order to, isolate those stem cells. Because see, the earlier two stages are lost. Blastocyst is when the fertilization occurred, right? So that is going to be no more, right? You are already an individual. You're not in that stage. Umbilical cord, a chance event. If your parents have preserved it, if they had the money and time to do that, they have done it. Mostly these days, it is due to awareness and money. Even if you have the awareness, if I tell you you have to pay 1 lakh as a lifetime fee to store an umbilical cord, if you are from a middle class background or a lower middle class background or slightly, I do not need to generalize anybody, but basically, in short, if you can't bear the cost, then you will not opt for it. You will just say that it's okay, you can cut off and discard. But people who are rich may probably go for this. Others who cannot afford it will not have that option. So the only option left over here is your red bone marrow cells. And as I said, stem cells, they have got huge applications in the industry. A lot of things are being done in modern science. There is a branch which works on stem cells, which is known as iPSCs. So that is nothing but induced pluripotent stem cells. So pluripotency is induced in those cells due to a lot of other things like physical stress, radiation, random mutations, certain drugs that stimulate pluripotency, growth factors, etc., etc. So pluripotency is forcibly induced into the cell. So that is also taking place. There has been an in-text box in your chapter about INSTEM Bangalore and NCCS in Pune. So NCCS in Pune was also established on these backgrounds and highlights to do cell biology research, to do biological research. There are certain scientists in NCCS who work on stem cells, cancer biology. So whenever you have some free time or you have your vacay after 10th, please go through these websites. If you want to do a career in pure sciences, it will be really helpful. Even if you do not want to, just as a curiosity matter or a science student, you can always sit and Google it. If you need aid of your teachers or parents, you're most welcome to have it. But please do have a look at these websites, INSTEM Bengaluru and NCCS Pune, and of course there are a lot of other institutes that have been mentioned in context-dependent manner in your textbook about the same. So about stem cells, there are of two major types. Okay, what are they? Embryonic and adult stem cells. So how do we differentiate or distinguish? As I told you, whenever there are such things, always remember them as differences and similarities. So remember, one side the opposite is the other. In biology, of course, the examples are different. They might not be exactly the opposite. So embryonic stem cells, as I said over here, they are present in the blastocyst stage or the early stages of cell development or the zygote development. So obviously, they are found only during early stage of blastocyst. That is where the example in which your embryonic cell cells are found is the blastocyst only. There is no other organ or organelle for it. And they are pluripotent, which means they can give rise to any lineage of cells that you subject them to, depending on the stimulus they receive. Then you have the adult stem cells, which are found over here, as I said, in the red bone marrow adipose tissue. So they are these undifferentiated cells in different organs or tissues. So undifferentiated means what? They are not differentiated yet. So there is a progenitor cell or a parent cell, and it can give rise to its specific lineage. That is what it is called as multipotent. So whenever the cell gives rise to a specific lineage, if I say the lymphoid branch of the immune system, so with the progenitor cell will give rise to lymphoid branch, the lymphoid branch of cells, that is NK cells, T cells, etc. Right? So these are your lymphoid cells. So it will have the potential to generate any cells, but from the same lineage. It is like trying to clone anybody, but within the family. Now, if you want to clone it beyond the family, then you have to have them from this stage, that is the embryonic stage. So coming back to adult stem cells, they are multipotent as converse to the idea of pluripotency in embryonic stem cells. The examples are 5 to 6, please remember all of them, preferably. Bone marrow, your brain, your liver, blood, skin, skeletal muscle and adipose tissue. So all of them at certain points and regions do contain these multipotent adult stem cells. That is why I told you they can still be derivatized from an adult individual. But as I said, if you derive them from the skin, it will give rise to cells of the skin lineage. You cannot make blood from it. If you derive them from the red bone marrow or from the blood, maybe you can synthesize white blood cells or RBCs from it, but not otherwise. Okay? So, after this, we have got applications of stem cells. So, I write them down as a diagram. So, we are talking about applications of stem cell biology. So the two major applications, as obvious they might seem, is your stem cell therapy for certain genetic disorders or other mishaps that you might have occurred which are congenital with any defects which you have since birth or otherwise. And then a major application which is still found use is your organ transplant. So you can regenerate either those organs in vitro or tissues and then transplant, or then you can carry out certain transplantations even when you are alive. So the transplantation can be carried out in alive as well as moribund or post-death individuals. Moribund is the brain dead concept. Okay, it is a medical term which you may or may not remember. So to begin with, the organ transplant, whenever a person is alive, it can donate certain parts of these organs which do harbor your stem cells. I told you adult stem cells are found in your skin, blood and liver as well. So liver, partially you cannot donate the entire liver, but partial donations are possible. So this is not given in a textbook, but this is a fact. Some organ or some portion of your liver can still be donated. Of course, for any of these donations, the blood group matching is utmost important. So whenever a person is alive, the skin, blood, kidney and liver can be transplanted. So what is the thing about kidneys? A person has got a pair of kidneys. So ideally, he or she can survive even on one of the pair. So if you can donate the other, but then it is of course a risk. If tomorrow that kidney fails or it becomes dysfunctional, whatever, then you might need a kidney donation or a dialysis as a treatment modality. But nonetheless, if it is required, if the person is willing, you can always donate any of these 4 organs. Then, post-mortem or post-death, you can donate certain organs completely. What are they? They are your liver, heart and the eyes. So, especially for brain dead people, so the person is alive, but the brain is dysfunctional. So, even if he lives life from, he will not understand you get up, he will not recognize you because he's brain dead. It is as good as his heart, that is what rather. His heart is pumping, but his brain is not working. So such people can actually donate organs in their life, or they can be put to sleep. They can be euthanized with genuine permissions, if at all, and then you can completely transfer these organs to the required individuals, to the accepting individuals who need them. But of course, there are a lot of regulatory mechanisms, legal issues and matters that need to be followed. Otherwise, there have been cases in the newspaper wherein people have actually been jailed or they have been put into courts to answer them why did they carry out certain experiments unethically. So certain things are not allowed. Every of these is very stringent and strictly regulated in state of development. So this was about stem cells. Now coming to your biotechnology applications, there are 5 major things that we will be studying. First is your fermentation technology, next is your vaccine and antibiotic development, DNA and protein products, GM bacteria, that is genetically modified bacteria, and your non-genetic or cell and tissue based methods. Okay, so let us briefly see what all of these are with certain examples. So fermentation technology has been covered in your introduction to microbiology. You tell me now, what do you understand by fermentation technology? Curdling of milk, it is a day-to-day activity in our homes. Production of cheese, liquor, beer, all of these are fermented products, isn't it? So they form a part of the fermentation technology. For vaccines and antibiotics, if you have seen the movie Vaccine War, I urge you to see, not of course at the cost of your exams and studies, but a very well crafted movie. So you'll understand what vaccines are and what Indian scientists did at that stage of time, where we were fighting the Wuhan virus infection. I mean, it will actually bring tears, or you'll be deeply moved by the efforts that scientists took during that stage for vaccine development and the upscaling of it. So vaccines and antibiotics are certain compounds which are made by bacteria and fungi to deter the other bacteria or fungi. So they usually kill them. Antibiotics primarily and majorly were produced, or rather still are produced from actinomycetes, which are predatory bacteria. So they have this intrinsic quality of predation. So they will try and kill other organisms and eat them. Eat them as in, this is a very layman concept, but they will try and keep them at bay. So basically they will prevent competition. On the other hand, they also derivatize some nutrition and food from them. So antibiotics are your fungi and bacteria that produce antibacterial compounds. Best examples are your Penicillin notatum from the fungi. Antibiotics, you have streptomycin, then you have got so many bacteriocins, polymyxins, neomycin, so on and so forth, which are produced by bacteria itself, which act on the other group of bacteria. And then you have the DNA and protein products. Again, this can be linked with vaccines. You have got mRNA vaccines. Then you have got certain peptide-based vaccines or toxoid-based vaccines. So the TT that you take, the tetanus toxoid, so it is again an application of biotechnology, wherein it is a toxoid which is generated in vitro. It is not the organism itself, right? For polio and other things, if you see, we have the live attenuated vaccines. So what do you mean by live attenuated? Attenuation is you lower the strength. So the organism is capable of dividing and causing you harm, but it is weakened due to heat, shock, or certain chemicals like formaldehyde. And that is why it is incapable of giving you a full-blown infection. But nonetheless, if it relapses or reverts, then you can actually be infected with the disease. So on sporadic occasions, there have been certain instances where people who took the polio vaccine got infected with polio for life. But that's very rare. So that is about DNA and protein products, which are advantages over classical live vaccines or live attenuated vaccines. And GM bacteria, genetically modified bacteria, which have got so many uses, and we come into them in details. The best example is insulin resistance that people have. So diabetes people who take subcutaneous insulin, right, before or after meals. So that insulin that they take is produced from E. coli, which is a bacterium. So that has been genetically modified to produce a protein or this hormone that is insulin, which is of human origin, right? How can a bacteria otherwise produce something of human origin? And it does not need it, right? So this has been introduced. Genes have been introduced into the organism synthetically, due to which it produces insulin. And of course, there are other growth hormones, growth factors also, which I will be coming to them in details as per the context. And then you have these non-genetic or non-invasive methods. Simply it is cell or tissue based, which is like somatic hybridization, or then you have your tissue grafting, like simply you, Alfonso mango, best example, what in Marathi we call kalam karne. So, twig of Alfonso mango will be taken and it will be grafted on the rooted plant of alphonsoid. So that is how they say that the flavor enhances or the fruiting is of a better quality, the yield increases, so on and so forth. So this is grafting with reference to plants. But animals, if there is a skin burn, right? The burn patient wards, don't ever go there. You will have a nightmare. Actually, people who are 70-80% burnt, they literally look very dreadful. You cannot bear the sight. So such people are grafted with human skin, which is generated or developed, cultured in vitro. Again, in vitro means in flasks or some plasticware. So these are non-genetic methods which are again put into anthropological use. So now after this, we will be seeing each one of them in finer details. Welcome back, class. So now let us see what are the commercial applications of these biotechnology methods that we are using. So welcome back, class. Now let us see the commercial applications to biotechnology that we have to study in this chapter. So first is your crop biotech. So in crop biotechnology, you have these three types with the examples listed below. So first is hybrid seeds and fruit. So obviously they are of a better flavor, they yield better, and even their yield in total as a crop is also high, and they also fetch you a good market price. So the examples of the best example is your seedless grapes. So seedless grapes, they don't have seeds. The classical white type grapes do have seeds in them. But now here you see the drawback is every time you have to sow the seed, you have to go to the vendor or the company who made this. Usually they have got monopolies. You don't have the seed, how will you sow the plant again? Because that is not through stem propagation. It is not a hibiscus plant or some other flowering species like rose wherein you have some grafting and then it will grow. It doesn't work that way. So they have got their own limitations and pros and cons. So everything should not be, started in a jiffy. Oh wow, you're getting so much of yield and so good, it's fetching me money. That is for one season. After you sell your grapes for one season, where is the seed to resow? Isn't it? So everything comes with some cons. So this is about your hybrid seeds and fruit. There are many to this. There are certain hybrid fruits also which are being produced, right? Which you will know better. You can Google it out. And the second thing and the most important, which has found profound interest in the Indian agriculture industry and the worldwide, is your genetically modified crops. So what are they? They are usually BT resistant cotton and brinjal varieties, which are very common. We eat normally what we eat is also a BT resistant brinjal. Okay, in Marathi what we call harkacha vanga or bharla vanga, the veggie that you make. So BT is Bacillus thuringiensis. So I told you in the previous classes that this is a type of toxin which is produced by this bacterium known as Bacillus thuringiensis. Now this bacterial toxin gets activated in the alkaline gut of the worms, the bollworms that feed on these cotton and brinjal crops. So you see those wriggly creatures, creepy corollis, the caterpillars. So they have got an alkaline gut. So when they consume the plant parts of these parts like cotton or the brinjal fruit, the toxin gets activated in their guts and it perforates it. So it will lead to a leaky gut and the organism will die eventually. So but it has got no harm to us because we do not have an alkaline gut, so they do not cause a problem when we eat them. And besides, those organisms or parasites eat the plants raw. We cook them. First of all, we do not eat cotton. Have you seen anybody eating cotton? No, right? Brinjal also, we have no dish which lets us consume, at least the Indian way of cooking I'm talking about, that you have it raw. You will either make it like a curry or you make kap or legume bhaja as we call. So you process the food before eating. So many times this is inactivated and owing to the later fact we do not have alkaline gut as the caterpillar has. So this toxin has got no effect. If that would be the case, obviously the crop wouldn't be in market, right? That is a fact. Then you have something known as golden rice. Now golden rice is going to produce beta carotene, which is a precursor to vitamin A. So vitamin A precursor is going to be produced. This was first synthesized and put into use through the Japanese culture of the Japanese farming because they actually had some severe vitamin A deficiency. So that is about BT cotton and golden rice. And of course, you have got herbicide varieties of crops, so on and so forth. Then you have this important thing of biofertilizers. So biofertilizers are nothing but a nitrogen fixing bacteria. I told you rhizobium is a symbiotic organism. So it needs the rhizomes, it needs the roots to reside. That is an anaerobic way of fixing nitrogen. Hence, since it is associated with rhizoids, hence the name is rhizobium. Rhizoids means something pertaining to the roots of the plant. So the root nodules that you see on legumes like matki, harbara, if you uproot the entire plant, you'll see the roots have got certain nodulations. So these nodulations are the home or the residence of this rhizobium bacteria. So it is not a free living nitrogen fixer. These organisms, Mastop, Azotobacter, Anabina, a type of algae, they are all free living. So under aerobic conditions also they can fix the nitrogen, which is advantageous, isn't it? You can just spray suspension or put the powdered formulation and then still get a biofertilizer out of it. Otherwise, you have to give NPK. Typically, all of these farmers give NPK. NPK stands for nitrogen, potassium, and phosphorus. Namely, NPK, N, P, and K. This stands for nitrogen. P stands for phosphorus. And K is for kalium. Kalium is nothing but your potassium. That is potassium. So this is usually given 30:30:30, or the ratios differ depending on whether it's a flowering plant or a fruiting plant. So this is about your crop biotechnology. Coming to animal husbandry, there are two major modes. One is artificial insemination and embryo transfer. Embryo transfer is similar to what we studied in life processes part 2. So you will carry out some IVF or in vitro fertilization, and then you will implant the embryo into the uterus of the cattle or whatever animal you're trying to breed. And then there is artificial insemination. So many a times what happens is you cannot physically let the animals breed owing to their differences in species or the size. Many a times, if the male is too huge and large, which is the case, it might lead to fractures of the females in case of cows. Because these days people are mad with the getting high amounts of milk that we were getting. All the milk that we drink is from Jersey cows, right? And there is a lot of hype these days about the A1, A2 milk, right? So the one produced by Desi cows or the ones which are inbreed, the intrinsic breeds which India had, like the Gir, Thalpakar, or in Maharashtra you have certain other breeds as well, like Hinari, etc. So these are indigenous breeds which actually yield much lower amounts of milk as compared to Jersey cows. But then, thinking from the perspective of the farmer, he cannot survive on such cattle, so they are resorting to what are Jersey cows. So Jersey cows is an example of how you obtain the offspring from an Indian parent, the Indian female. So this is through artificial insemination. So I told you then that there are semen banks. So the veterinary doctor actually gets a semen vial and he inseminates the receptive female, which leads to hybrid animal formation. Then coming to human health, there are huge applications to it. I can go on for hours like this. But then, just to begin with, we have these two major things, that is vaccine, disease diagnosis, and vaccine and vaccination or vaccine development. So disease diagnosis is your various kits that are put into use, the pharmaceutical applications to these drugs, like you take for people who have got hypertension, they have a lot, depending on what is your range, you take a lot of drugs. So these drugs can either be produced synthetically or there can be certain monitoring or modulatory mechanisms through which you can diagnose the disease. So various kits. You remember during the Wuhan virus outbreak recently, we even had got this MyLab kit of home testing, Covishield, not Covishield, that's the name of the vaccine. So you could self-test if you have COVID. So all pregnancy tests. All these kits and tests are applications of biotechnology. So either they will have some anti-sera or antibodies immobilized onto them, which will give you the test, or there will be some chromogenic test, just as you have the pregnancy test. And then you have got this important aspect of vaccines and vaccination. So vaccination is the process of you or any individual getting vaccinated. Now, how are they produced? In an industry, of course, Serum, best example in India, the largest vaccine producer throughout the world. They produce a number of vaccines which are viral based. There has been some hype about mRNA based vaccine. So tetanus toxoid is an example of vaccine that you have to take every 6 months, especially you take them prior to rainy season because you get scratched, you get bruises, and then you don't want it to become septic. And why do you don't want to get it becoming septic? Because sepsis will cause anaerobic conditions in it, in which Clostridium tetani will thrive. I told you then that Clostridium is a obligate anaerobe, which means it requires no oxygen to survive. And then you can get contracted with tetanus, a deadly disease, which is incurable. So you take this TT every 6 months or whenever you have a deep-seated cold. mRNA-based vaccine, they already have shed enough light on them. Then talking about an interesting thing of edible vaccines. So edible vaccines are produced in this transgenic potatoes, which will give you immunity against E. coli and Vibrio cholerae. But here the thing is you have to consume it raw. Again, because processing is going to denature the vaccine, it is going to render it useless. And you tell me when do we have it raw? So the applications are limited. Will you eat a raw potato or will you go for French fries? Obviously the latter, isn't it? So there are applications to it, but then there are certain limitations owing to these things, like the way in which we have the acceptance to it. Like voluntarily you will never have a raw potato, right? But if you have this transgenic potato which bears immunity against E. coli and Vibrio cholerae, you may be protected. Now there are few more other applications which we will be seeing as a flow diagram. So moving ahead, let us see what are the other applications to treatments of human diseases with reference to applications, industrial applications of biotech. So first is your hormone production. Insulin, somatotropin, growth hormone, all of these are genetically introduced into plasmids which are then introduced into E. coli. Okay, so this E. coli is a bacterium which is going to produce and synthesize these peptides or molecules, precisely hormones, for our intervention and our use. So what you do is you let the organisms grow, then you lyse them, that you burst open the cells, and you after certain processes of isolation and purification, you get the purified products, which are of course subjected to certain quality assurance and quality control checks before they are marketed. So as I told you, the subcute insulin vial that you get, the bulb, is actually produced through E. coli. And earlier times, why am I laying so much of emphasis on it is, during earlier times it was known as a disease of the royal. Only the royal people could afford treatment to diabetes, the others were left to perish. So in first, pancreas of horses and pigs were utilized to get this insulin. So they were of course sacrificed. There is no way you can remove live pancreas, like what will the organism do then? So diabetes is a type of autoimmune disorder. Just as a passing point, I am telling this to you. So your beta cells in pancreas which actually produce insulin are degenerated. They become dysfunctional due to degeneration, and hence you have no more insulin in the body. So coming back to my point, initially it was harvested from such macroorganisms. So they were killed. You know how expensive that is and how inhuman. So now due to advent of modern technology, we are able to isolate this gene from beta cells of the pancreas which produce insulin, which has been introduced into the bacterium, right? And then this bacterium is capable of giving you a purified product, of course with certain processing, which is obvious downstream of for isolation. Then you have got this interferon producing E. coli. Now you would say, why is there E. coli everywhere? There are so many millions of bacteria, but E. coli has found maximum usage owing to certain advantages that it has got a very short doubling time. So 20 minutes is the doubling time. So in 20 minutes you will get 2 cells from one cell. Then you will get 4. It is logarithmic. So on and so forth. It is easy to handle as well. Not easy to handle, I would say, but due to a number of factors, especially the doubling time, the yield, and the genome of E. coli is also well mapped and well studied, and culturing them is easier. Again, which what I meant by handling is easier. It does not have very rich media requirements like mycobacterium has. So certain organisms need very specific growth media, but E. coli is typically a middle class organism which is very easy to grow. It will grow in algae broth, like urea broth or nutrient broth, very easy. So interferons are molecules which interfere with the viral replication. Whenever you have a viral load or a viral infection, the cells are going to resist the infection of the virus or the virus entry into the cell. And the molecules that do this are known as interferons. So interferons are going to inhibit the viral entry into the host cell. So this interferon producing E. coli is also an application to it. Then you have got gene therapy. So gene therapy is when you introduce certain genes on purpose. So an example quoted in your text is for PKO, that is phenylketonuria. So that is a disease condition which leads to coloration in the urine because proteins cannot be digested. So this leads to phenylketonuria, and then the treatment modalities for it are different because you actually is no drug which can treat PKO. So then you go for gene therapy. The details are not expected from you at this stage. Just remember what gene therapy is used for. It is used for treating PKO. What is PKO? What is alkaptonuria? What is phenylketonuria? What amino acid is responsible for it? Knowledge is used, but it depends on how much you want to acquire, how much is essential. So please stick to it. Then you have what is cloning. So in cloning, you have got two types. You have got therapeutic cloning and you have got your general cloning or somatic hybridization. The somatic hybridization, in short, is nothing but fertilization without, per se, your sperm cell. So what you do is you take two cells, okay, one cell is enucleate, the other is multinucleus. So this is the enucleate cell. Now you remove the nucleus from this cell, you isolate the nucleus, and then you fuse it with the cell A, let us say, and this is cell B. So this fusion of the nucleus from one cell into the enucleate cell into the other enucleate cell, which happens to be A in our case, is known as somatic hybridization. Right? So in somatic hybridization, there is no need for two gametes. The fusion of the nucleus with the enucleate cell is capable of giving rise to a complete cell lineage, a cell, an organ, or even an organism altogether. The best example of cloning is your Dolly, the first sheep that was cloned. So this was also through a enucleate cell cloning process. So a nucleus from the female was taken. It was fused with the somatic cell of the other, the donor pair, the ovum cell, and then that was implanted into the uterus of a female. And then the offspring that was born, she was a female sheep, and she was named as Dolly. Before Dolly, it was done with frog. Amphibians are easy to work. So there are a lot of examples in history you'll find about cloning. So this happened without mating or copulation, without the involvement or fusion of two different gametes. Hence, we call it as somatic hybridization. So nucleus of one cell is fused with the enucleate cell of the other, and this can give rise to an entire individual. So this cell, okay, this is not a normal cell. This is your egg cell, and the nucleus will be from your somatic cell, a non-sex cell. So it is known as somatic hybridization. Now coming to other applications, I will give you a flow chart. So just wait for it. So coming back to our other applications, we have these four major ones again. That is your industrial biotech, environmental biotech, food biotech, and DNA fingerprinting. So in industrial biotechnology, we had this what is known as the white biotechnology, in which yeast are genetically modified to exclusively produce only ethanol and of course the side product, that is carbon dioxide and water, from molasses. Ideally, yeast have this quality, but they have been specifically made or genetically engineered or modified to give you ethanol in higher amounts from molasses, which are substrates for alcohol production. So molasses is a waste product from the sugarcane industry. Then in your environmental biotechnology, you have got a number of examples enlisted in your textbook. Please go through them. Microbes and plants used for cleanup. So this is how I sum it up. So whenever you have got certain toxins or heavy metal or effluent built up in the environment and you want to clean it, but that is not physically possible for you to teach, you put other organisms to use in a smart way. So here, The water hyacinth or Jalaparani as we call in Marathi or in Hindi. So you will see usually it is a green colored plant covering the. So then talking about environmental biotechnology as I was talking about, I told you that microbes and plants are used for this cleanup. So what do I mean by cleanup? So as I said, whenever there is some toxin or other products that build up in the environment, we put organisms other than humans into play. So we put them to work. So as I was telling, water hyacinth, which is known as Jalaparani, is put into use to soak up heavy metals like lead, cadmium, arsenic, which are let into the water bodies due to industrial effluents. So these plants are known to absorb heavy metals and store them. So this is more done by a process known as bioaccumulation, right? So just remember this one. Then the mustard variety is also engineered to remove selenium deposits from soil. So soil which is highly contaminated in selenium deposits, you can crop these plants over there and then you can have them removed from the soil. Similarly, Deinococcus radiodurans, which is a very famous example of a microbe which is intrinsically resistant to a high amount of radiation in terms of grays or rads, in the units of measurement for measuring radiation. So this has actually been put into use since it absorbs a lot of radiation. It is being utilized to absorb radiation from radiation contaminated soils or environments. So this organism otherwise is probably harmful, is not harmful. So it is being put into use similar to lead. Analogically, our radiation chambers or radiation materials is always housed in a lead box or a lead cage. Why? Because lead or plumbum, Pb as the symbol for it in the modern long form of periodic table, is capable of absorbing a lot of radiation. So this organism is a live entity and it can absorb a lot of radiation. So radiation contaminated soils are being spread or being populated with this organism to absorb radiation from them. Then food biotechnology, I will skip this point because I have enough given you examples. The examples are your cheese, curdling of milk, making paneer, yogurt. So all these applications where you put into use different strains of microorganisms, lactic acid bacteria, or in certain cases fungi to impart flavor to cheese, is your food biotech. Then coming to DNA fingerprinting. DNA fingerprinting, the institutional example given in your text is of CDFT Hyderabad. So this is done to solve cases of criminal or forensic origin and to understand or a certain inheritance. So if I claim that somebody is my child, how do I claim it? So there should be some portion of DNA which should be common. The technique is known as RFPD, okay? RFPD, RFLPD, whatever. So there are different techniques which we utilize for DNA fingerprinting, like RAPD is rapidly amplified polymorphic DNA. So those DNA bases amplified should match between me and the offspring whom I am claiming to be mine, or the other way around, whoever is claiming somebody to be their parent. So these techniques are done through DNA fingerprinting, where you actually do exact matching. So they need to be identical. Okay, identical means exactly same. Similar is 99% is also considered similar. But identical means it should be a 100% match. Okay, so they need to be identical, and the uses are these I told you in forensics and in order to solve cases of inheritance. Then coming to revolutions. These three major revolutions are very prominent and again very well understood with the set of examples. As I told you, this entire chapter is example based. You remember the example and you will be able to explain the concept. So the Green Revolution was started by Sir M.S. Swaminathan in India and his US counterpart, Dr. Gordon. So they together devised the strategy for mass production of crops, precisely the dwarf wheat variety, which was resistant to a lot of biotic and biotic stress factors. Biotic factors are your pests on crops, like the bollworm I told you. Then the abiotic factors are your environmental factors like drought, less rain, excess rain, wind, etc. So these crops were made with a high yielding output, and they were dwarfs, so they were less subject. So anything that is tall, see, if I am of an average weight, if I was 6 feet tall, I would have problems sitting in a car, right? If there is a huge wind blowing, I will have more resistance to if there, on the contrary, if there is somebody who is he will easily get it somewhere. Obviously, he or she will have their own set of pros and cons. But otherwise, the dwarf plants are advantageous as compared to tall plants when it comes to mass plantations in the field. Then, white revolution was started by Sir Verghese Kurian. He recently passed off a few years ago. So this was through the milk cooperation movement in Anand in Gujarat. Amul is a wonderful miracle that we see out of it. So he started this for milk production, and India is one of the leading producers of milk in the world, and we also export, apart from the fact that we also have to import. But milk production was then increased to several times high as compared to what it was. Now, green and white revolutions were started in the 20th century owing to the population explosion that India was facing. Of course, we got independence. We were not completely independent in terms of food. The best example is when we imported grains like wheat from other countries. We even got the parasite and invasive species with it, the Congress grass that you call, isn't it? So, such were the problems associated. So, these two visionaries, Sir Verghese Kurian for White Revolution and Dr. M.S. Swaminathan for Green Revolution. Please remember their names because they have contributed a lot to our generation and even the previous generation wherein our parents were approximately our age. And that has led to India being the foremost in terms of grain and milk production. Then recently the government launched this NKM16, that is Neel Kranti Mission 16 in the year 2016, which is termed as Blue Revolution. So why was this term coined as Blue Revolution? Because it was done with an intention to produce the aquatic produce or increase the aquatic produce that one can get in terms of fish, shrimp, and many other oysters, etc., which can be of high value even in terms of food as well as other aesthetic purposes. So these were the three revolutions, or rather are the three revolutions that we are still underway through. Please remember them and they may be asked for your short notes and other important questions. So now after this, there is one short point of your grain or farm produce processing, organic farming and fertilizers, which I'll be telling you about. So nearing the end of this chapter, we have got these 6 subpoints to study. So the first one is hydroponics, which is a new term to you. So what do I mean by hydroponics? It is basically culturing or cultivating plants in a soil-less fashion. So you always know that plants need a substratum to attach, which is soil. Okay. So what here we are going to do is, we are going to culture plants devoid of soil. So basically it is just going to be your water and you will be cultivating them on a liquid substrate. So you have to give them some physical support for the plant to rest, but you will not require soil. Now this is of great advantage in today's time where we are facing a lot of soil erosion and a lot of farming land being converted into any that is non-agricultural land for industrial purposes and building blocks and societies or turning them into various different schemes, because humans are falling. There is explosion. A result of explosion is we need space to stay. There is less space. So a lot of farmers, if the land is barren, it is fine. But these days, even agrarian societies of farmers are getting converted their non-agricultural land to non-agricultural land through certain certifications, which is not right. And then they are getting money out of it. But this is partial. After a period of time, when we will be divorced, see, you can't make soil, you could construct buildings. But if I tell you to make mountains and I tell you to make soil, will you be able to compound? Nobody has ever been able to do that, isn't it? So hydroponics is a concept which is of relevance in today's time owing to lack of soil and space. So a lot of research is being carried out in this. Then you have got fertilizers. So fertilizers, I mean, you should be using natural sources. Okay. So your natural sources are what? Cow dung, or then you have your produce or the ones vermiculture or vermicompost. So this should be preferred over non-biological or inorganic fertilizers like NPK. NPK gives you a good yield, no doubt, but after a few years the soil becomes saline or it becomes infertile. So every crop you have to do this. But with this, what happens is you regenerate the capacity of the soil to give you a good yield. So fertilizers preferably use biological, that is best are cow dung and your vermicompost. And of course, you can use, coming to our revising our previous concepts, you can use nitrogen fixers as well, like azotobacter and ammonium. I told you, these liquid suspensions are available which you could spray and then you could naturally get your nitrogen fixed. Then coming to insecticides, a lot of chemical insecticides, malathion, DDT, etc. have proven to be harmful to higher organisms, owing to bioaccumulation, and they have also led to disease resistance. The best example is DDT resistant mosquitoes. Now you see, mosquitoes don't die of DDT. Why? Because it was so rampantly used, and I told you, evolution is a continuous process. The host also evolves and the parasite also evolves. So in this case, mosquito is our parasite, we are the host. So we put out chemicals to kill them, they evolved ways to resist. So insecticides, it is better to introduce or improvise on the plant immunity in terms of plant biotechnology rather than spraying chemicals. DDT, malathion, all these are chemicals, right? They are insecticides, chemical insecticides. Then what are the biological interventions? DDT cotton is the best example. I told you take a natural gene from an organism which encodes for a toxin. That is one way. The other method is you can use certain organic class of compounds which trigger immunity in plants. So if you do a lot of research, you will find that a lot of research has been going on in this field in order to intrinsically enhance the immunity of the plant. Then you have got apiculture. So apiculture and these three things are like side businesses to your mainstream. Okay. Apiculture is your cultivation of bees, honey bees. So the honey, these days you get them of many flavors and they have got their own medicinal uses as to from which flower is the honey prepared. So honey bees collect nectar. Okay, this nectaries are then processed in their hives, which gives you honey. Plants do not have honey. Please get the terms right. So that is about agriculture. Then medicinal plant cultivation. Apart from your mainland farming, you can also culture and cultivate certain medicinally important plants like Tinospora cordifolia, gulve. So it does not need a lot of space. Aloe vera, best example. From the Thar desert, like Thar aloe vera. You get brands these days which sell out aloe vera juice saying that it is from Thar. So aloe vera needs very less amount of nurturing and care. It goes in less amount of soil. It is a succulent species. A less amount of water, I'm sorry. So that is one aspect. The last aspect that we are studying over here in this chapter is the fruit process. So this also you'll find traditionally the roots of this have already been laid in our culture. If you go to Konkan area in Maharashtra, you'll see in these days that households are busy preparing what? Three things. Amba poli, fanas poli, and the amsola that we say. Amsola is nothing but the sour fruit that we utilize for making solpad. So that, because there is ample of sunlight in the coastal areas, the area is humid and very dry. So amba poli, fanas poli are what? They are applications of the fruit pulp which is produced and stored for a long time. So when seasonal fruits come once a year, jackfruit and mango are the best examples. So what do you feel like eating them later? Will you go and store them in liquid nitrogen tanks? Right? So you process it in certain ways by either drying or sweetening it more, so that it adds to its shelf life, so that it extends. Shelf life is increasing the extending the or increasing the expiry date of certain food products. So by increasing the shelf life, you are adding to its storage value. See, milk. Milk is a very, very perishable item. Now, if you make ice cream or if you make curd, that will still have a better shelf life. If you store the milk packet in your freezer, that is at around minus 20 degrees, it gets stored for a lot of period of time. So what are you doing here essentially? You are increasing the shelf life. You pasteurize it to Tetra Pak. You are increasing the shelf life. That is why you see the Amul Tetra cans or any other company, Mother Dairy or whatever brands that you get these days. So they are packed in Tetra aluminum sheets. You get those boxes, right? So they are sterile. The milk remains outside the fridge for 6 months. That is what they claim, and that is true. If it is spoiled, it will go puffed, it will go stale. But that is meant by processing. So that is with reference to milk, no doubt, but even fruit pulp. You get mango shakes all around the year. How do you get it? You have those mango pulps, right? Whenever you feel like having some mango smoothie or mango shake or mango shira, mom will always have this fruit pulp which she has made in-house, or then she can get it from places outside, right? So the bottled or the canned mango juice is an example of fruit processing. So this brings us to an end of this chapter, which is very extensive. And as I told you, please remember it with examples so that it is easier for you to understand and write answers. Conceptually, there isn't much to understand. This is all an example-based chapter that we are studying. I hope you enjoyed learning this. Until then, see you next time. Goodbye.